and also the drug development. Basically, the drug development principles are similar across human health and animal health, and however, the way it is done in different organizations and different countries, and the way it is looked at from different regulatory authorities is slightly different. And CMC is nothing but chemistry manufacturing controls. It's part of the drug development where it deals with the drug substance and drug product, right? And so together it is called drug development and CMC. While drug development is very specific to early stage for preclinical until the phase one studies, the CMC is actually at different phases. So the chemistry manufacturing controls is at the early phase, it is early phase CMC, then the late phase CMC. Even after the drug is commercialized, you will still have the CMC part. But what we focus today is only on the early part of the drug development. So, and thanks to Bhagya for providing some introduction. So, though I am a core pre-formulation and formulation scientist, I would like to call myself as a scientist rather than anything else. I like to actually call myself as more as a pharmaceutical development leader, where I don't even only represent the drug product or pre-formulation. So it's actually the combination of everything, and that's actually fascinates me of taking this role. And Elanco Animal Health is actually Eli Lilly's Animal Health division. So after working nearly for 19 years in human health, now I transformed myself into animal health industry, where the drug development principles are same, but there are new learnings. So the learning and the other side of the world that I wanted to explore was the reason why actually I took this opportunity and seriously considering to continue for the rest of the life. So let's go into the topic. So what we'll do, as we don't have a lot of time today, Bhagya has given me only 1 hour. And so we will briefly see how the drug discovery and early phase drug development process works, and then the contribution by the chemistry colleagues, formulation colleagues, and safety assessment. Our next speaker, Mahati, is going to actually talk more about that. If there are any mistakes, please forgive me, Mahati. Clinical drug product, analytical development, regulatory authorities, then the actual clinical study, and then how all of these functions come together in an integrated model. Right? So I always emphasize on this integrated approach, be it in your personal life or your professional life. The intention and involvement drives the end result. So this is not a new picture for anybody. Everybody knows that it starts from 10,000 compounds and it goes to one approved medicine. And what we are going to do today is not focus so much here and here, but on the early phase drug development or CMC, where the compound is transitioning from drug discovery to early development, and that early development includes safety in humans. So the objective of all the drug development companies or the regulatory authorities is whether the drug is safe. That is the first criteria. No regulatory authority will really look into whether this drug is efficacious or not. They don't really care unless you prove that it is safe. So the safety is the first and foremost important thing. And once you establish the safety, then only they will give you the permission or approval to actually go and test the actual hypothesis. And so the early phase drug development constitutes of many functions as we see, and then finally we see only one approved medicine. So another important thing to note here is it's not always that I discovered the drug and it should be in the market. So the way you should look at it is, I am discovering this drug, is it worthwhile to bring this into the market so that tomorrow if I am a patient, can I take this? So unless you are prepared to kill the molecule, you see here the negative is the positive. The early you kill, the better for the good candidate to move forward, and it also saves cost, resources, time, everything. Right? So it's not always the positive thing that is positive, even the negative thing is a positive in drug development. And like every activity, so there is a syllabus, right? So I am also taking the syllabus here, where since you cannot cover everything, so we are currently focusing on small molecule drug development. While I don't know anything about the vaccines or large molecules, how they work, so since I have only worked here, I can only share that much to you. And oral route of administration. Again, it's a different topic. Mostly, I have worked on the oral route of administration and enhancing its viability and so on with different dosage forms, and not so much in the other dosage forms. And CMC strategy for early phase targeting phase 1 clinical trial. So what we talk, everything will be related to how we are advancing the compound faster to the phase 1 clinical trial. Why it should be faster? Because regulatory authorities are providing that leverage that you don't need a huge package, point number one. And the second thing is you cannot spend too much time in the early phase, because unless you test in human, you don't know whether it advances. So taking a lot of time before going into human will not help the organizations, and they save more money to actually invent more drugs. So that's why the speed is in essence for early phase drug development. Of course, regulatory authorities also will help here. We'll talk about that little later. So the most important thing is how the compound transitions from NCE, transition of NCE from discovery to development. So I will not use a word API in the entire presentation, because API is something which is after the drug is approved. So it is not an active pharmaceutical ingredient, it is just a new chemical entity. And either otherwise you can call it as a drug substance and not API. So that's the difference between the API and the drug substance or the compound. So basically, as we know, we have two different phases. Now, discovery, we know there are different stages: target selection, hit to screen, thousands of compounds, hundreds of compounds, dozens of compounds, and then maybe 2 to 3 compounds in lead optimization. So there are several clinical candidates. Every drug discovery scientist thinks that all of these can become drugs, but which is not the truth. So then you actually measure this or evaluate these candidates, clinical candidates, so-called clinical candidates, with respect to physicochemical properties, biopharmaceutical properties, and safety aspects. While you don't do a full-fledged studies during the drug discovery, but it is very important that we have the knowledge on the physicochemical properties, biopharmaceutical properties, nothing but the PK and dose proportionality, and then the safety, whether it is really safe or not at the discovery stage. And so these candidates, the clinical candidates, so-called, let's say there are 2 or 3 clinical candidates, and you subject them to these processes, and there is only a chance for 1 or 2 compounds here, they have to pass this gate. So just like the entry of the college gate, they cannot allow everyone. They allowed me because one student accompanied today. So the developability assessment gate. So this is the stage gate where lot of discovery compounds are screened at different stages, and finally when you have a confusion that I have 3 clinical candidates and only 1 or 2 should go, then it has to go through. And then in the drug development or early phase CMC, as you know, there is a drug substance development. Initially in the medicinal chemistry it will be few milligrams, and then here you may need few grams to kilograms to really support the program. So we'll talk about each of these little later. Then the preclinical studies, where it also includes the GLP toxicology study. Whenever it comes to G, it is good laboratory practice, where the quality assurance is involved. Then we have the drug product development, where what is the suitable formulation for preclinical and what is the good formulation for phase 1. It may not be the final one which goes into the market. You may see that in the market it is tablet, but actually in the preclinical study it could be a suspension or solution. And then analytical methods, we should know what is the assay of this API, what is the residual solvent, what is the assay and dissolution of this drug product. So analytical methods, not only from the from those perspective, but also from the spectroscopy and structural characterization. And then you manufacture the clinical batch under GMP, and then you prepare the regulatory filing. So you submit the regulatory dossier to the authorities. There are several regulatory authorities that we will discuss, and then finally the first in human or phase 1 or clinical study. So actually all these three are kind of synonyms. First in human or phase 1 study or clinical study, just so that everybody are well versed with a particular terminology. I thought of listing all the three here. So this is the overall process of how the drug transitions from discovery to development. So let's see the drug substance development. And so the main thing is the synthesis here. In the medicinal chemistry, usually it happens that even if you get 100 milligrams of the compound by employing 1 liter of solvent, it is fine, because ultimately your objective is to get some material. But imagine if you want to make 1 kilo, you cannot put 10,000 liters of solvent, right? So the medicinal chemistry objective is different, and when it comes to development, there the drug substance development objective is different. So here you need to really concentrate on the synthesis, not on the compound. Of course, final output is the compound, but on the synthesis. And also, you should be very careful in selecting the reagents, because many reagents are hazardous. They are known hazards, and we should avoid. You can also see that in the ICH guidelines there are different classes of materials, different classes of solvents that you can use, and then there are limits for them. So the more hazard material you use, the difficult it is to remove and then qualify your material according to the regulatory. And no intermediates with toxicity, whether it is toxicity or genotoxicity, and the selection of the route, selection of the solvent, and the yield is also important. And then you also need to see what could be the scale-up challenges. Then once you set up your synthesis or the route of synthesis, then you see what are the properties that are required for the formulator or for the toxicology. See, basically the chemical development or the chemistry department, for discovery, chemistry is the customer. For chemistry, toxicology and formulator is the customer. For toxicology, clinical is the customer. So we actually look at the customer. So that means you are in a service industry. So whenever you are in a service industry, you have to do a good job. So target properties, now you know when you should decide whether you need salt or free base based on physicochemical properties. Then the surface or bulk properties, so wettability and things like that. Particle size distribution, you know the impact of that. Impurities, tox and FIH batch, because you cannot have a new impurity in your phase 1 batch because you have not qualified that in the toxicology study. And then finally, the quality and quantity. So it's not only the quantity, but also the quality which is important. So typically it goes like this. Initially about 25 grams, then 200 grams, then 1 to 2 kg of tox batch, and then 2 to 4 kg of GMP batch. This is typically what happens in the drug discovery and development industry. So let's talk a little bit on the formulation development. So formulation development is actually in 2 phases here. It's not just the formulation that goes into human, because unless you have a good preclinical formulation, you don't understand how it behaves in the animal and what type of formulation is required, whether it's a simple solution or suspension is sufficient, or you need to go to enabling formulation like solid dispersion or nanoparticles, things like that. Now these are the typical animal models. So the rodent models and the non-rodent models. And first you need to understand the purpose of the study. So though the job is to do the formulation, unless you understand what is required for the preclinical biology or pharmacology group, then you cannot do a good job. So whether it is a PK study, whether it is a dose range finding study before the toxicology, that is a 28-day GLP repeat dose study. I think Mahati will cover a lot, she is the expert here. I will not talk much about that. So animal models, whether it is rodents or non-rodents, again rat, mice, guinea pig, dog, mini pig and monkey, because the route of administration also is important, hence the excipient selection will become different and the dose volumes will become different, and animals also will behave differently with different excipients. Now duration, whether it is acute study, chronic study, or whether you want to dose it once or twice, because if it is an acute study, you can actually use nasty excipients and then see the do the exploratory study. If it is a chronic study, then there will be a vehicle effects, and rather than looking at drug effects, we need to concentrate on the vehicle effects, which is not good. And then the excipients, permissible daily exposure, it is not only for humans, it is there for even animals. You need to be very sure of which excipient for which duration of study and which animal species and route. For example, surfactants cannot be administered as intravenous in dogs, it will die immediately of convulsions. So there are several criteria that how we select the excipients even for preclinical formulation. So exposure, whether it shows a good bioavailability or bioavailability enhancement is required. Sometimes what happens, it may show a good bioavailability, it may have a dose limitations where vomiting and diarrhea may be seen, and then you may have to divide the doses between BIT and QD. Then dosage form, is it a simple suspension with unmicronized compound, or is it something I need to micronize, or we need to do some enabling formulation, solid dispersions, etc. And then we always do not have success every time, right? So there are some compounds which are unstable in solid and solution state. So no matter how you try, so the compound is not stable, there is not much a formulator can do here. And very low permeability, you cannot use a lot of high permeability enhancers because they are all not regulatory wise very attractive or acceptable. Then high clearance, you can make the drug enter into the blood, but if it is immediately clearing off, then there is no question of bioavailability and first pass effect, pH effect, food effect. So these are some things which are out of our control. These are some of the things which we need to consider before we go into the preclinical formulation. Now, once I have the preclinical formulation, I will directly not go into the clinical formulation development because I need some doses. So what strengths I need to do, and for that the safety assessment, even before the safety assessment you need to do the PK study, because you should know the doses, up to what dose you see the dose proportional exposure, and with what formulation you can see the dose proportional exposure, and then how do you achieve the objective of the safety assessment, which is identifying no observed effect level, no observed adverse effect level, and maximum tolerable dose. Mahati can correct me in the next session if I have done anything wrong here. And then these are the rodent models and non-rodent models. And basically, you also select the disease models, which happens much more earlier stage. But here, by the time you reach the safety tox stage, you actually know which species and then the dose range finding study. Before the actual 28-day GLP repeat dose study, you actually do the dose range finding study and find out up to what doses the animal can tolerate. So why these are important, right? NOEL, AEL and FTD. NOEL is nothing but no observed effect level, where you don't see literally any pharmacological activity or toxicology, and that is the safest dose in animals. We'll talk about why safest dose in animal is required, and then why MTD, maximum tolerable dose. So according to the regulatory requirement, you have to characterize the toxicity. So if you say that my 3 doses or 4 doses I selected is safe, then your package will be rejected. So you have to show that there is a toxicology for this compound, or your dose should be 2 grams per kg. And imagine if it is 2 grams per kg, then people have to eat that in the plate. So it is not possible. So hence that's the way the regulatory authority controls how you develop the drugs. And this MPD. So basically now because humans are privileged species on the earth, so we don't want to make them experimental animals. So all these are under our control, so we can use them and then leverage that knowledge and data, and then go to the human, so that we can kill the animals, but we cannot kill the human. So now, before you actually go and develop your formulation, let's say I want to make a tablet or I want to make a capsule, so I should know what is the projected clinical dose. Not only the therapeutic, we are not talking about therapeutic dose here, it's only the safety. So whether it is safe or not, if you have to show it whether it is safe or not, then you need to give ascending doses, escalating doses. If you have to do that, then you need to know what is the safe dose and you need to know what is the maximum tolerable dose. The same concept applies to human also. There is a FDA guideline in terms of how you need to calculate the human equivalent dose or maximum recommended starting dose. You see, it's only the recommended starting dose. It still did not say what would be my final dose. Now what happens? You determine the NOEL which is there and convert each animal NOEL to human equivalent dose, and then select this HED from most appropriate species. In the sense, for example, if the NOEL is lower for dog, then you consider that rather than the rat, because you have to talk about safety. That's all. And then choose the safety factor divided by HED by that factor, because safety factor is something like, for example, if NOEL is 10 milligrams per kg, and at 100 milligrams per kg you still don't see the mortality or toxicity, then the margin of safety is 10 times. So that factor you can divide it here, and then you will arrive at MRSD, which is the maximum recommended starting dose. So as a thumb rule, if the NOEL in animal is 10 milligrams per kg, most likely the starting dose for human would be either 1 milligram per kg or 0.5 milligrams per kg. It can also be derived based on body surface area, not only body weight. And then you also consider other factors which are where there is a physiologically active compounds. Then there are other factors which will come into picture. Now, one tool which is known as allometric scaling, and it will provide the derived pK parameters from the pK parameters of different animal species. Like, so where you have concentration time profiles of these and the AUC and Cmax and clearance and volume of distribution parameters, you can translate and calculate, estimate that for human. So the pK parameter against the body weight or body surface area. So there is a very good tool called allometric tool. So you just cross check that whether this allometric tool and the concept of MSRD is same or not. So, and now I know that yes, these are the doses for the formulator to come out with a formulation for clinical, and now the drug product development. So basically the most important thing is physicochemical properties. Unless you know about the drug, you cannot really develop a dosage form. And polymorphism, size, particle size, shape, solubility, solution stability, hygroscopicity, bulk density, wettability. You need to have the knowledge and understanding of the impact of that on the both manufacturability as well as in vivo performance should be understood. And biopharmaceutical properties, check what is the absolute and relative viability, whether it has a dose proportional viability or not, and need of enabling formulation. Consult your BCS expert here and see whether we need any enabling formulation or not. And then look at the clinical requirements, whether it is a blinding versus open label study, what is the clinical dose range, whether a placebo is required or not, and the timelines. So always we talk about fit to clinic and fit for purpose formulation. We'll see little more about this fit for purpose formulation. There are some very simple methods that you can still use and dose it to humans. Then the limited quantity of API, you will not have kilograms and tons together like paracetamol to explore. So you have very limited amount of API and you need to come out with simple formulation and time. And then finally the regulatory requirements, whether we need a GMP product or GMP like, mostly it is GMP, and what is the requirement for filing. Just a time check again. Okay, good. So some of the 20 minutes are left or 20 minutes are left? Okay, sorry. So dosage form types generally used in phase 1 clinical trials. So drug in capsule, you actually just add the drug inside the capsule. You don't mix with any excipients, but definitely that also requires some kind of knowledge. That it should not be hygroscopic, it should not have gelling tendency. And drug in bottle, you just put the drug in the bottle, send it to the clinic. The clinician will add some kind of liquid, a marketed flavored liquid, and he can administer. Amorphous solid dispersion when you require higher bioavailability. Powder blend in capsule, which is a classical capsule formulation. Tablet, immediate release or controlled release. This amorphous solid dispersion powder also can be converted into tablet. Then pellets, as is in a capsule, whether it is immediate release or controlled release is not very common in phase 1, but pellets. Liquid in capsule is again when you have oils and you want to fill the oil into the capsule. There are liquid filled hard gelatin capsules available. Not exactly the soft gels, but even in the hard gelatin. And then some of the formulations like lipidic excipients, you just dissolve the drug, pour a molten solution into the capsule, and then it will solidify. So several options are available. These takes less time, less effort, low cost, because you need only 1000 or 2000 units for clinical and stability. So then the analytical development, I need to know how much I put in the whether I have really added the API in the product or not. Then these are the people who are going to tell me. And at the API stage, the spectroscopic and chromatographic methods are very important. The NMR, MOS, LCMS or IR. You need to really come out with a structural elucidation and say that this is my structure. Single crystal PXRD is also very common. And bioanalytical methods for really analyzing the blood samples. And then one impurity method for drug substance and drug product, because you know exactly which impurity you are talking about. When the analytical method is different for drug substance and drug product, then again it will pose a challenge saying that whether this impurity is the same what we are talking in drug substance or not, because all the toxicology studies are done with API, clinical studies are done with formulation. So then the force degradation to understand the degradation mechanism. So the drug substance methods for intermediates and final compound like carbon, hydrogen, nitrogen analysis, spectroscopic analysis, the HPLC analysis, residual solvents and heavy metals, particle size and solid state with XRD, DSC, ATGA, etc. Impurities and counter-ion content if it is a salt. So for the drug product, assay and content uniformity and blend uniformity, you study in textbook. Impurities, water content, dissolution is the main thing. Dissolution here is basically a QC tool. It's not going to correlate between dissolution and human exposure because we don't have human exposure yet. So it's only about the QC release criteria of dissolution and microbial testing. Whenever you are administering any drug substance or drug product to human, microbial testing is a must because we don't know if the compound or excipients have any microbial contamination. So now, why we are doing all these things? Actually, Dr. Arun Kumar also mentioned that we have to satisfy the regulators. So finally, you and me can think this is the best, but regulator will say no, this is not how you need to do. So for this, they also give an opportunity to talk to them. Like for example, FDA will say pre-IND meeting before you submit the IND. You can actually say this is my strategy, this is my limited data, these are my thoughts. They say okay, go ahead, or they will give a suggestion. So that after submitting the regulatory dossier, instead of getting the queries and then delaying your phase 1 program, it's always better to consult them in advance. Then I start with India, CDSCO, it's also called IND. In USA it is FDA and it's also called IND. IND is the IND filing, which will give permission to go to humans. This consists of drug substance, drug product, the biology, efficacy, pharmacokinetics, toxicology, everything in a package. And then in Europe it is the European Medicines Agency. It is called Investigational medicinal product development. UK it is MHRA, but again it is called LMPD. Japan it is PMDA, again it is called IND. In Australia it is known as clinical trial application, where therapeutic goods and administration is the regulatory body, and Brazil it is ANVISA. I could not find a relevant one, but the format should be as a common technical document format. Now, what the regulatory authorities, because it is early phase, so you need not have huge set of data and experimentation and proof, like a process validation of three batches stability and all, because you cannot spend so much time and money when you don't know whether the drug really advances from phase 1 to phase 2. So hence, they provide you the opportunity to make small batches. They say don't do any process optimization or anything scalability studies, because that's not relevant at this phase. Full validation of analytical methods is not required, only do some kind of specificity, linearity and accuracy. No need to go for robustness, inter-day precision, intra-day precision, different labs, different people. So all those things are not required, which is required for later phase. And 1 month stability is sufficient for drug substance and drug product. And you need to keep on updating your data with the regulatory authority. And eventually you have to submit 2 or 3 years. But to start your to submit your application, you just need 1 month stability, and then they will give approval within 30 days. Most of the regulatory authorities give approval within 30 days, so that you don't make the drug substance and drug product and wait for 6 months to really administer the for humans. So these are some of the things that you can leverage with the regulatory authorities and advance your drug faster. So now the first in human study or phase 1 clinical study, again, I don't claim that I know a lot here, because the role is to understand what they need and provide the drug product to them in the dosage strengths that are required. But there are 2 types majorly: the placebo controlled clinical trials, where there is a control or placebo group and the experimental or treatment group. And here the requirement for formulatory is you need to have placebo and different formulation strengths. Whereas in the blinded versus open label study, so here non-blinded, usually it happens for anti-cancer and some of the high therapeutic areas where you cannot really use a placebo. So you cannot use a placebo for a cardiovascular drug and say that yeah, now the person is right and it's working. So that's not allowed. And there is only non-blinded study. Double-blinded, both physician and the participant does not know. In single-blinded, only the participant does not know. But the main criteria here is, if you don't know this in the beginning, then you may have different looking placebo and different strengths of formulation, like 10 milligrams